Thank you very much for inviting me here. It's been 2 spectacular days, and it's, of course, a daunting task to follow Duško's very impressive presentation here, focusing on the role of the gut microbiota in humans. So what I will focus my talk on today is then on the mechanistic, or some mechanistic studies, mostly being involved in using mouse models. But I would like to start with a slide that was something that Alan just brought up, is that over the past decades there's been a dramatic reduction in infectious diseases, whereas at the same time you've seen an increase in autoimmune diseases especially. And that's been proposed to be something called the hygiene hypothesis that I'm sure you're well aware of, that perhaps that we're living in a too clean environment. And that's been sort of suggested by Bengt Björksten just a couple of years ago, that maybe we should call it the microbial deficiency syndrome, that we're missing some microbes early in life that really is aiding us into developing into a healthy human individual. And what we heard just a couple of minutes ago, and was also published by Peter Turnbaugh, is that there's a reduced microbial diversity in obese subjects. So perhaps there's a microbial deficiency also in obesity and metabolic diseases. So this is also to highlight that something that sort of is very hot now. It wasn't hot maybe 10 years ago. So Josh Lederberg, the Nobel laureate, published a perspective paper in Science 11 years ago. And I think this is really cool because he had a perspective that we should start to focus on the metagenome. And what he wrote was that we are a host of a poorly cataloged ensemble of symbionts to which we pay scant attention, yet they are equally part of the superorganism's genome with which we engage the rest of the biosphere. And this is exactly what Duško just presented, that we need a catalog of what we have in the human gut. However, also that although we know what we have there, does not necessarily tell us what the gut microbes are doing. So we've had a lot of backgrounds here, so I will skip over most of it. But what we would like to do is to view the gut metagenome, or the gut microbiome, or microbiota, as an organ by itself, present in the human gut, with the size of the liver weighing 1.5 kilograms, with several different cell types or bacterial species, with a fantastic potential to affect our metabolism. And the gut microbes can do that, and I've just grouped a couple of functions here, that they have trophic functions, such as they control epithelial cell proliferation, which was something that Bruno Lemaître talked extensively about yesterday. And I will show you some data on how the gut microbes can promote intestinal angiogenesis. And of course, the gut microbes have a very big potential of educating the immune system, which has been the focus of most of this colloquia. The gut microbes also have protective effects, in that they can protect against pathogens. We heard also about that yesterday from Brett and others. But I will focus then the rest of the talk on metabolic functions of the gut microbiota, how they can affect our metabolism. So the technology we use is germ-free mice, and we had some background to that yesterday, but I just want to point out that it's nothing new. The first attempt to have germ-free animals was actually over 115 years ago in Germany, where they tried to make germ-free guinea pigs. They weren't successful. They died from infections after 13 days, but it shows that this has been an interest for a long, long time. And when James Reniers, some 75 years ago, managed to get the first cohorts of germ-free guinea pigs, that's really starting an era, which maybe slowed down during the 70s and 80s, but now picking up again. And this is just to highlight that in 1948 you could get a thesis on describing how to rear germ-free rats, and that was Bengt Gustafsson in Sweden, who has had that as a thesis project. And one of the hallmarks of a germ-free animal is a very extended cecum, as compared to from a conventional raised mouse, or if you colonize a germ-free mouse for just 2 weeks, you will shrink the cecum into normal size. So of course we need to keep the mice sterile. Over time we use a slightly different system that Andrew McPherson demonstrated yesterday. We also have flexible film isolators, but they are more sort of custom made. We have them in different sizes and shapes. But the general idea is that we filter the air on its way in and its way out. We bring in sterile food and water and bedding and instruments through this port on the back here. And then we have the mice in regular cages. So I would like to tell you first a little story about how the gut microbes can affect angiogenesis. And this is something that's been demonstrated by Jeff Gordon's laboratory 10 years ago, where they compared germ-free and conventional animals, and could demonstrate that the conventional animals had a much more developed blood vasculature system in the small intestine. So I had the great pleasure of having a super talented postdoc, Christoph Reinhardt, who's now leading his own research group in Mainz, who joined the lab, and he had an interest in further delineating the mechanism by which the gut microbes can affect angiogenesis. So what Christoph did first was to just demonstrate that you had a much more developed blood vasculature system by staining for PCAM1 or CD31, which is a vascular marker in a conventional raised animal, or compared with a germ-free animal. We could also colonize a germ-free animal at adulthood for 2 weeks and reconstitute the blood vasculature network. So this is quantified here. We can also do that by qPCR. We can qPCR analyze for different vascular markers as VEGFR1, as well as a potential mediator of angiogenesis, angiopoietin-1, which is also increased in conventional raised animals. Christoph then isolated epithelial cells, which should be the ones that are in contact with the gut microbes, and could demonstrate that the epithelial cells had a significant increase in angiopoietin-1 expression in conventional raised animals compared with the germ-free animals. So to test whether angiopoietin-1 actually was a mediator of angiogenesis, what Christoph did was that he took germ-free mice, he treated them with an angiopoietin-1 inhibitor, and then colonized them, kept on treating them every 3-4 days for 2 weeks, and then sacrificed the animals and analyzed them. And what he found was that actually treatment with an angiopoietin-1 inhibitor dramatically reduced the angiogenesis in the small intestine. We could also demonstrate by, once again, taking out the small intestine, we could demonstrate that we had increased phosphorylation of Tie2 in conventional raised animals, demonstrating increased activity of angiopoietin-1 signaling, which binds to Tie2. So in other words, we think that the gut microbes induce angiopoietin-1 in the epithelium, which then mediates increased angiogenesis. So how can this signal be going on? So Christoph did his thesis work with Bernd Engelmann in Munich and worked a lot on tissue factor. And tissue factor is a very interesting molecule. It's a membrane-bound receptor that is mostly known for its activation of homeostasis via thrombin activation. But it's also known to be induced in angiogenesis either by direct signaling or via activation of proteinase-activated receptors. So Christoph set up an experiment where he treated germ-free animals, similar to what we did with angiopoietin-1 treatment, with an antibody against tissue factor. And then he colonized the animals, and then he kept on treating the animals until he killed the mice after 14 days. And what Christoph found was that treatment with an anti-tissue factor antibody significantly reduced the amount of angiogenesis, here as from staining with PCAM1 or by qPCR for PCAM1, another vascular marker, VEGFR1, as well as angiopoietin-1, suggesting that tissue factor is upstream of angiopoietin-1 production. To test whether tissue factor was important, he used mice in collaboration with Wolfram Roof at Scripps in La Jolla that were deficient in the cytoplasmatic tail of tissue factor. So these are conventional raised mice that are just like the wild type, except that they lack 20 amino acids in the cytoplasmatic tail of tissue factor. And quite strikingly, these mice had a reduction in angiogenesis. Furthermore, we could demonstrate that tissue factor is phosphorylated on this domain if we colonize the mice. So to test whether proteinase activated receptors were involved in the signaling cascade, we first analyzed by qPCR the levels of PAR1 and PAR2, which has been the mostly described in terms of angiogenesis. And we could see that PAR1 was significantly increased upon colonization, whereas PAR2 was not significantly different. So we then analyzed the angiogenesis in PAR1 or PAR2 knockouts by staining for PCAM1 again, and we could demonstrate that PAR1 deficient animals had reduced intestinal angiogenesis compared to wild type, whereas PAR2 was not significantly different. That's also validated by PCAM1 mRNA analysis And we could also demonstrate that PAR1 deficient animals had a reduction in angiopoietin 1. And what was really striking for us was that if you knock out PAR1, you do not any longer phosphorylate tissue factor. So all this data and a lot of other in vitro data led up to a model that Christoph proposed, that we have the gut microbiota in the lumen of the intestine, which induces several glycosyltransferases that induce glycosylation of tissue factor, and I didn't have time to show you that data. And this glycosylation then moves tissue factor out on the cell surface, where it activates factor 10 to 10A to thrombine, and thrombine can then activate PAR1, which then phosphorylates tissue factor. So this is sort of a feed-forward loop where you have tissue factor activating its own phosphorylation cascade. Phosphorylation of tissue factor then leads into angiopoietin 1 expression, which mediates angiogenesis. So this is an example on how we can use animal models to get at different mechanisms and combine with either pharmacological treatment or by looking at knockout animals. So I think also this angiogenesis is probably a response to the colonization, and that we have a germ-free animal, and I don't know if you noticed that, but the villi is very thin and elongated, probably to maximize the absorptive capacity. But when you colonize the animal, you will have, of course, an infiltration of lymphocytes, but you also have a shortening of the villus and a widening of the villus structure, and that really requires more oxygenation, so you have more angiogenesis. And what we recently found is that if we block tissue factor and angiogenesis, we also prevent the widening of the villus. So I will now move on to the second part of my talk, where we will discuss some of the metabolic functions induced by the gut microbiota. So what we did is a quite straightforward experiment when I arrived as a postdoc in Jeff Gordon's laboratory, where we said that if the gut microbiota really affects adiposity, we should be able to measure that by comparing the amount of body fat in germ-free and conventional raised animals. So we did that using a DEXA, or dual energy X-ray absorptiometry, where we put the mouse into a machine and we get a number of how much fat it contains. So what we saw was that the germ-free mice had about 8% body fat, compared with the conventional raised mice that had about 12% body fat. And if we colonized these germ-free mice for 2 weeks with a normal gut microbiota and measured on the same mice 2 weeks later, we could demonstrate that we had about a 50% increase in body fat in as little as 2 weeks. The phenotype is quite evident. So if we open up a germ-free mouse, we see that the fat pads are much smaller compared to a conventional raised animal or a mouse that's been colonized for 2 weeks at adulthood. The effect is really rapid. We see it in as little as 10 days. We see it in males and females, and we've seen it in several different mouse strains. So I think this demonstrates that it's really powerful if we can combine the metagenomics with some functional studies in mice, where we here can show that the gut microbes indeed can affect body composition. So how would the gut microbes go about this? And one of the first hypotheses we had was that the gut microbes may affect energy harvest due to its capacity to ferment polysaccharides. So in an experiment that was conducted by Peter Turnbaugh in Jeff Gordon's laboratory, they sequenced the metagenome of a few obese mice and demonstrated that they had an enrichment of polysaccharide degrading enzymes. And indeed that the gut microbiota of the obese animals had an increased propensity of harvest energy from the diet as measured by remaining energy in the feces. This also corresponded with increased levels of acetate and butyrate, which are the end products of bacterial fermentation in the cecum. And importantly, if you then transplanted the microbiotas from the obese or the lean mice into germ-free C57 mice and analyzed them 2 weeks later with DEXA, he demonstrated that the obese microbiota induced significantly more adiposity as compared with the wild-type microbiota. And we think that a lot of this is working, is a correct hypothesis, but because when we analyze the amount of body triglycerides in the liver of germ-free and conventionalized animals, so these are mice that have been colonized for 2 weeks, we could demonstrate that these colonizations increases dramatically the amount of triglycerides in the liver. It's about twofold more if we quantify it biochemically. So that further supports that the
colonization of the gut microbes will increase the amount of energy inflow into the liver, induce lipogenesis, and increase triglycerides. And these are then stored into the adipocytes, which are enlarged in the conventionalized and conventionally raised animals compared with the germ-free counterparts. So in order to further look at what are the lipid composition of the adipose and liver in germ-free conventionally raised animals, together with Matteo Oresic's group in Finland, we performed lipidomics analysis comparing germ-free conventionally raised animals. And here is the data from the adipose tissue. If we looked at the liver, there was about 100 different triglyceride species that were increased upon colonization, corroborating our previous stainings of triglycerides. But it was quite interesting when we looked into the adipose tissue. There was about 16 lipids that were altered, and 15 of them were triglycerides. And most of them were, as expected, increased in conventionally raised animals here compared with the germ-free animals. And the ones that were increased had even carbon lengths. So they were most likely derived from de novo lipogenesis when you add 2 carbons at a time. Whereas the 2 triglycerides that were odd-chained, they were increased in germ-free animals, suggesting that these 2 probably derived from the diet. So that led us to propose to a potential mechanism where in the gut we have a lot of gut microbes, of course, that can then generate short-chain fatty acid that reaches the liver through the portal vein. We've also shown that colonization will lead to increased blood glucose, which also can contribute to the increased lipogenesis. This then leads to increased VLDL production and increased incorporation into adipose tissue through an lipoprotein lipase mediated mechanism that is driven by fatty acid-induced adipose factor. So it's an inhibitor of this enzyme that's reduced by the gut microbiota. Furthermore, we demonstrated that upon colonization, you also reduce fatty acid oxidation. So you promote the synthesis of lipids and you reduce the consumption of lipids, which then potentially could explain the increased adiposity. So if life was that simple, if we would take germ-free animals and then feed them a diet which is rich in sucrose and lard, which is fat, we would expect that these mice would develop obesity. So we did that experiment, and to our surprise, we could see that the germ-free animals did not at all gain the same amount of body weight when fed this high-fat, high-sugar diet as compared to the conventionalized counterparts. And this has now been confirmed in 3 different studies, so we really think that this is the case. However, I should point out, and we'll come back to this, that according to Fleisner et al. In Michael Blaut's laboratory, it seems to be dependent on the macronutrient composition, whether the germ-free mice are protected against diet-induced obesity or not. So then we had to say that, well, this has probably nothing to do with energy harvest from the gut or from the diet, since the germ-free mice should be quite proficient in taking up sucrose and also triglycerides. And in some recent experiments where we've looked into metabolic inflammation, which is associated with obesity, where we basically stain for macrophages in adipose tissue, we could demonstrate that in conventional raised mice, in the adipose tissue, we see significantly more of these crown-like structures that are aggregates of 13 to 15 macrophages surrounding a dying adipocyte. And we see definitely significantly fewer of these in the germ-free animals. We can quantify them. And then we, of course, ask a question. Is this due to that the macrophages in conventional animals are deficient in responding to LPS? So we isolated macrophages from the intraperitoneal cavity and then stimulated them with LPS in vitro. And we could demonstrate that both macrophages from germ-free and conventional raised animals responded equally well to LPS with a junk phosphorylation. So we don't think it's the macrophages per se that do not respond to whatever stimuli, but rather it's a deficiency of migrating into the adipose tissue. And that's demonstrated here that the mice macrophages isolated from colonized animals have increased TCR2 expression, which is a chemokine receptor involved in macrophage migration into the adipose tissue. And we're currently doing some in vitro experiments that seems to substantiate this, but we need to validate those experiments. So increased metabolic inflammation in the adipose tissues, and
associated with reduced insulin signaling. And indeed, if we compare the insulin tolerance of a germ-free or conventional raised animal, which we do by injecting them with insulin and then measuring how rapidly the glucose is reduced, we could see that the germ-free mice respond much better to this insulin injection, which has their blood glucose drops more and it remains lower for a longer time as compared with the conventional raised animals. So if I should summarize where we are now, it's that we have started to realize that the gut microbes affect our metabolism, but they do that by several different mechanisms that could be totally independent or dependent on each other, such as that we have harvest of short-chain fatty acids that can act as substrate for lipogenesis, but we also have pro-inflammatory molecules such as LPS that could drive metabolic inflammation in the adipose tissue. We have regulation of fatty acid oxidation, and we have regulation also of signaling molecules in the small intestinal epithelium, such as fatty-induced adipose factor, that then suppresses. So if you suppress PF as you do in colonized animal, you will relieve the inhibition of LPL, and you will have more triglycerides that are stored into the adipose tissue. And I think that we will see much more of these, as Sven showed earlier, that we probably have microbial regulation of eating centers in the hypothalamus, etc. So I'll just finish with, I think, a fascinating paper that was published a month ago in Nature, which illustrates that it's really important to look at the interaction between the diet and the microbiota. And in this particular paper, they were interested in microbial metabolism of phosphatidylcholines, which are present in high amounts in high-fat diets or in high-fat foods that we eat as humans as well. And phosphatidylcholine are then metabolized in the small intestine, and by the gut flora, you will have the emergence of 3-methylamines, which then is converted in the liver to 3-methylamine N oxides. That can lead, as I will show you, to heart disease. So what Wang and his co-workers did was at first that they looked at the covariation between these metabolites, choline, TMAO, betaine, and the odds ratio for cardiovascular disease. And you can see a quite dramatic odds ratio with 8, 16, 8-fold. And to demonstrate that this actually has these food components has a role in developing heart disease, they fed mice either with a choline-rich diet or with TMAO, and they could see that you have a quite significant increase in the atherosclerotic lesion size. So this just shows that if you feed these metabolites, you will have any increased risk of atherosclerosis. So what they did then was that they did the same experiment, but now they treated the mice with antibiotics. So if you have a controlled diet, which is rather low in choline, you have no atherosclerosis. And if you treat them with antibiotics, you don't see a significant difference. If you instead feed with choline, you have an increase of atherosclerosis, but if you do that in the presence of antibiotics, you have a significant reduction of the atherosclerosis. And that's quantified here. You see it in females, you also see it in males. And that this choline feeding also increased the amount of macrophages in this atherosclerotic lesions. So, for me at least, this is quite evident that you have an effect of the gut microbes only if you have certain diets. And we've done a similar experiment where we look at development of atherosclerosis in mice, and we use ApoE mice, which we read-write as germ-free. And ApoE mice have a propensity to develop atherosclerosis. And what we find is that if we look at males, we can find germ-free males that don't have any atherosclerosis, or with a reduced amount of atherosclerosis compared to conventional raised counterparts, and that's quantified here. So we have some mice that do not develop any atherosclerosis, some that do develop a reduced amount of atherosclerosis. However, in females, we don't see that at all. And quite strikingly, the diet that we've been using up to now has 0.2% choline, and that's sort of in between that experiment I showed you previously. So we're now trying to figure out whether this is actually a choline-dependent mechanism or not. The atherosclerosis in conventional raised males is associated with then increased plasma cholesterol as well as increased levels of KC, TNF-alpha, and RANTES. So I would just like to finish by a conclusion here that I think that the human gut microbiota may be considered an environmental factor that functions on the intersection between diet and our genes to affect physiology and metabolism. I think that will be sort of adding on to the complexity as discussed before, that we're mainly talking about our genome and the microbes, but we probably have to take into account also the diet here. And I would also like to point out that what we're doing is nothing new. This is from Milwaukee Chronicle, 1936. Upon the publication of James Renier's findings, where they wrote in here that the question is whether humanity would be better off or worse off without any wicked little bugs. So I think that's still the question we ask today, and we're trying to understand the mechanisms. Of course, I didn't do the work. This is the group who's been behind all the work I presented today. And also, this is from our retreat, and we invited Patrice Carny, who's been working as a postdoc in Remens lab. And he contributed a lot to our studies. Should also acknowledge the work by Jeff Gordon, who's been very supportive over the years. Wolfram Roof for the angiogenesis studies, Matej Oresic for the lipidomic studies. Björn Fagerberg is a clinician who's been helping us to collect material from patients with atherosclerosis. And Ruth and Rob has been helping with some metagenomics studies that I didn't have time to present today. So thank you very much.